Good morning, everyone. Welcome to the Decode Science BGI STOmics Breakfast Workshop here at the Multiomics 2022 conference. I'm Richard Harrison, Director of Business Development at MGI, and I'll be your MC for this session. I'm sure you'll agree, after a very successful day one agenda, that while this may be the last conference on the 2022 calendar, it's by no means the least, and for many people it's been well worth the trip to come up here. And in particular, we're thankful that so many people have come out early on a Monday morning to join this breakfast session. We really do appreciate your effort in coming along, and I'm hoping the catering is sufficient and keeps you well G'd up for the day. I'd like to point out we have a healthy presence of BGI and MGI people in the room and on the booth later. And I'd like to call out our great partners at Decode Science. You can see Joshua and Peter up there wearing company blue. That's great. So if you have any questions afterwards, you don't get your questions answered during these talks, please feel free to call us out at the booth and have a conversation. If you've forgotten, now's a good time to silence your phones. This is a breakfast session, so a little less formal, so if you do need to sneak out and top up your coffee and come back, that's perfectly okay. We have three short presentations for you this morning. Firstly, we'll have our application scientist, Kira Sun, who'll give you a short introduction to the field of STOmics and StereoSeq for those who may not be across it yet. And Kira will also give a few pointers on where the technology is going in the near future and how you can get involved. Then we'll move on to the main event with our guest speakers and early access partners for STOmics technology. There'll be Jennifer Correndi of the Harry Perkins Institute in Western Australia and Quan Nguyen of the Institute of Molecular Biosciences here in Brisbane. Quan's going to talk about his work using StereoSeq to investigate the aging process, and Jennifer is going to combine StereoSeq and another novel technology from JumpCode, also available through our Decode partners, that increases the resolving power of this already pretty powerful technology. We'll be keeping to time today as we have to be finished in time for the main agenda and give you time for a bio break beforehand, but I think we should still have plenty of time for questions. So let me bring on our first speaker. Dr. Kira Sun joined the not-for-profit BGI Research Institute as a senior researcher in 2017 after obtaining her PhD in biotechnology in Ghent University in Belgium. She's engaged in the development of novel high-throughput metagenome sequencing technologies and in research in taxonomic assignment and functional studies of diverse microbial samples in fields as wide as soil health and rainforest ecology. Kira also has a number of first author publications in leading journals such as Nature Communications, Plant Biotechnology Journal and the Journal of Fungi. Kira currently acts as the field application scientist supporting the applications of BGI spatial transcriptomics innovation in Australia and New Zealand. So Kira's talk today is titled An Accessible STOmics Workflow for Any Laboratory. Thank you, Kira.
Thank you for the nice introduction, Richard. So I will give a short background of our technology and the future applications and how you're going to join us. Like those journal presents the power of StereoSeq technology on the embryogenesis development, including many different kinds of species, and also the brain science, and also in the plant part, the study, of Arabidopsis leaf development, and we also have a lot of preprints in different areas like cancer research, and other area are coming out like in the near future. So those publications empowers the StereoSeq to facilitate and accelerate the research. Back to the principle of the technology that we have this technology based on our DNA nanobore patent arrays, and that they are randomly located on the silicon chip, which has customized size, and the biggest part is 16 centimeter by 16 centimeter, which means that you need a bigger cryostat to do the sections. But back to the back to the DNA nanoball, their distance between each DNA nanoball is 500 nanometer from center to center. That for a mammalia size of 10 micrometer, it contains 400 DNA nanoballs to do the capture racing of the whole transcriptome. So there's DNA oligos on the DNA nanoball to record the coordinate information. And after the first sequencing, the coordinate information was gained, and then it goes to the linkage of the poly-T and the UMI for initiate the capture probes. So our product is go with a chip that contains all those elements, and then when you receive the kit, people will start the lab work from the cryo state and like laying the tissue on the capturing chip, and after several steps that in wet lab, for instance, the permeabilization, reverse transcription, all the transcripts from the tissue section are captured and being pulling into one tube for library construction. So in the end, after experiment of about 1.5 days, you will get the whole transcriptome results like located in a single tube. And that one goes for sequencing, and in the end you will have the step 6 exhibited, the whole transcriptome profiling from an intact tissue section. And this is illustrate the experimental steps, but to save some time here today, you can also visit our website, which is STOmics Tech, to find out more details and the detailed protocols and the videos to guide you how to work through this. Like towards to the future, next year we will have the automation systems from the post-imaging to library preparation to save a lot of time and accelerate the speed of the experiment. So it can be expected with less than 30 minutes hands-on time of experimental process, and you can perform 24 chips processing capacities in 1 day, and the whole procedures will about 7 days end to end. Yeah. So the whole pipeline from for the STOSEC is not only about the STOmics kit, but it's also available from the BGI group, like the MGI, to provide the sequencing systems that we have. Like some of you already have the G400, and it has higher throughput from our DNB6 TNext systems. That you can also visit our MGI website to look for the details of the throughput of those different sequencers. And for downstream analysis of the data generated from that, we have our in-house developed SAW StereoSeq analysis workflow and the StereoPi. So the original idea to develop those pipelines is because that StereoSeq can generates so much amount of data that no other technologies can be compared. So we would like to have our own developed pipelines to facilitate to analyze those amount of data at single cell resolution. Right now, we have this technology applied on so many different species and tissues for human tissue and mouse tissue, and also other different animal models that we applied nearly like across all the organs. So it means that StereoSeq is very applicable for many different kinds of research of interest. And for plant species, we are also trying to establish a mature workflow to facilitate, but due to the difference of the plant tissues, that it also involves a lot of studying and developing of those pre-treatments. So yeah, like I previously mentioned, and this is our website, it has the product information and the resources of the protocols, the videos, and also the latest news. For instance, last month we have this grant thing to promote our technology, but you can get the latest news from the website as well. In the end, I would like to welcome people like join us, the Spatial Temporal Omics Consortium. I think the videos also introduce that where we're wanting to build this community to facilitate this area development and to have this joint force to push through more scientific like breakthroughs to benefit science and clinical users. I would like to have the update on the STOmics product that has been released. You can start to order that product. It will have the features of the chip on the slide, which is easily for handle, and it supports fresh frozen sample, and with upgraded chemistry that improves capture efficiency, like about almost a double capture efficiency compared to the publications that we published on Nature and Science. So more powerful of this product, and we have provided the cell segmentation algorithm to provide the single cell resolution analysis pipeline, and in the future we will have more powerful image progressing software that you will see that it's more visible and hands-on on those new updates. So finally, due to the timing, and I would like to thank you all for getting up, joining us at our breakfast workflow, and welcome to join our STOC consortium as well. Thank you.
Any questions for Kira? Okay, thanks, Kira. Okay, so now moving on to the main agenda with our guest speakers. Dr. Quan Nguyen is well known to many here and is a group leader at the Institute of Molecular Biosciences at the University of Queensland. He's leading the Genomics and Machine Learning Lab to study neuroinflammation and cancer immune cells at single cell resolution and within spatial morphological tissue context. His research interest is about revealing gene and cell regulators that determine the states of the complexity of cancer and neuronal ecosystems. Particularly, he is interested in quantifying cellular diversity and the dynamics of cell-to-cell interactions within the tissues to find ways to improve cancer diagnosis or cell type specific treatments or the immunoinflammation responses that cause neuronal disease. Dr. Quan's GML lab is also developing experimental technologies that enable large-scale profiling of spatial gene and protein expression, spatial omics, in a range of cancer tissues, focusing on brain and skin cancer and in mouse brain and spinal cord. Quan's talk is titled Building Spatial Multi-Organ Aging Clocks. If you'd like to welcome Quan to the stage.
Thank Richard for the nice introduction, and thank everybody for attending the morning session today. My talk will be about the aging research, and I think this is a very interesting area and topic that are very suitable for using spatial technology as well. So why we studying aging? Is it because soon we will have about more than 2 billion people that are older than 60 years old, and this is not a problem of a developed country, but it's also a problem of developing countries. For example, we see in this plot here the orange and green color showing the increase in the number of people that are older than 60 years old, and the increase rate is actually higher in low developed countries compared to the developed country. So it is a global issue. And aging is a main risk factor of many diseases. For example, for the cardiovascular disease, neurodegeneration disease, immune-related disease, metabolic disease, and cancer. And importantly, different organs in the human body cause different types of diseases. And the aging rate between different organs are also very different. We are especially interested in the question that how different organs age differently and how the communication between organs in the body can contribute to the aging process of the whole body system. And we ask which method would be most suitable to answer that kind of question. Then we decided to use spatial technology. And the reason for that is because we think that the cells in the organ actually interact with each other and they signal between cells. And which technology would be most suitable for studying cell-to-cell interaction. We believe that the spatial technology, by measuring all genes and by locating the neighboring cells, can allow us to identify the interaction within organ and also interaction between organs and see how that interaction affects the aging process. Eventually, we want to build different aging clocks for different organs. And here in this study, we extracted 5 different organs from mice. The idea of this is that if we have a match organ in one mouse, we could really link the aging process between organs to each other. And we applied 2 spatial technologies as the independent and complementary technologies, and we also use the single-cell RNA-seq data to validate the results. So with the help of Kira and with the collaboration with the BGI team, our lab led by 2 experimentalists in my team, Yufan and Albert, have successfully generated the spatial data for 5 different organs. And it is quite ordinary to have the optimization from one organ to the other organ because there's quite different conditions. For example, here I'm showing the kidney and the color we are seeing here is the cDNA signal from the tissue. So if it's brighter, that means that that's a better condition for the sequencing. And if it's too bright, that means that a diffusion process may happen. So in here we show that 15 minutes is the best permeabilization time for the kidney. And when we saw the results, it's really the kind of result that we expected for. So in here we see the color. Each color represents one cell type or one tissue type, and this is the kidney. So we can see clearly the single cell resolution, the 3 layers of the kidney. So the cortex here, and this is the medulla and inner medulla, and that's at the single cell resolution. We can also see the mixing between different color. That means that we can identify different cell types per layer of the kidney as well. And all of this is come from a data-driven way. When we look into the detail of the brain data, so we can see in general, this is the QC information where we have this is the number of genes per sample, so very consistent between samples. And then this is the number of reads per sample, so quite high number of reads per sample. And here I'm showing bin 80. That is the interesting part of this data that I will explain next. So because the resolution of the STOmics technology is 0.2 micrometer, so that means that we have the flexibility to look at the data at different resolution, at different tissue scale. So it can be range from single cell resolution like this. So that's bin 14. Bin 14 means that there are 14 bits per dimension, X and Y dimension, and that's equivalent to one single cells, the size of one single cells. But then we also have the flexibility to look at bigger tissue units. For example, we can go to bin 50, and this is the signal from bin 50. And if we want, we can also go to other higher size, for example, bin 200. And the benefit of this is surprisingly very interesting in terms of the data analysis, because not always we need to look at the single cell resolution. Sometimes we want to have a very clear signal of the tissue region, and here is what we can see. So, for example, this part of the brain here, if we use the single cell resolution, we do detect the signal at the single cell level. But if you want to look at the brain region, then maybe using bin 80 is giving us the better signal. And this is the clustering of the brain, so we can see this cluster, very distinct cluster for the dentate ridges, and that is again the data-driven way of identifying the region in the brain, and suggesting that this type of data are really sensitive, so that we can locate different sub area of the hippocampus. And the heat map here is showing the unsupervised way of finding the gene signature for different tissue region, and we can see that the signature can be very specific to different spatial location in the tissue. So that is a really impressive way of looking at the data and understand the tissue at different scale, from single cell to the tissue area. We perform the comparison between young and old mice across the 5 organs, and we could detect the genes that are highly expressed in the old or highly expressed in the young mice, and then we could perform like a standard pathway analysis. Interestingly, the ligand receptor interaction turned out to be the top enriched pathway in the old organs. And when we integrate two types of data, so here we integrated the Visium data and STOmics data, we could identify 86 genes that consistently upregulated between the two technology. And interestingly, among those 86 genes, the top genes in here could be validated by independent single cell data sets. For example, here we have a complementary factor 4B, so that's the top most upregulated in the aged mouse, and it's also the top upregulated from the single cell analysis. But what is the addition to this is not only do we know that these genes are upregulated, but it's also we could find the spatial location of where in the young brain that is expressed much more compared to in the aged brain. So that is so this is one example of the oxytocin genes, and then we can look at the example of that complementary 4B factor. So here in this case it's much higher in the age and lower in the young, and it's enriched for the fiber tract in the brain. So that is the compared to the single cell, that's the additional information we can get. And when we combine the 5 tissues together, we could identify a core set of genes that consistently upregulated in the aged mice compared to the old mice, and that upregulated genes is in here. The top one is a complementary factor 4B, so that's related to immune pathway that clear up the dead cells. The most interesting part of this study, when we apply the spatial data to analyze the aging process, is to study the communication between the cells. And we develop an algorithm where we can make the use of the neighborhood between the cells, so that we can test for the 2 cell types that are using 2 ligand and receptors as the mean of communication that are much higher than random pairs of genes and random pairs of cells. So, so that approach using spatial data for cell-to-cell interaction can reduce the false positive detection. That is the key limitation in terms of studying cell-to-cell interaction using the genomics data. And we could make the comparison between cell-to-cell interaction between the aged and young mice. And this is an example of looking at the pair of cells that interact much more in the age or much more in the young. So that is the current work that we have done so far, and this is an ongoing work. We're looking for more collaboration. So if you have a relevant samples, or if you are interested in working with us to add more tissues type, please reach to us, and we really welcome the collaboration. So we've shown that we apply the stereoseq for multiple organs, and the analysis comparing between the young and the aged reveal some of the biological processes, especially the complement pathways that are consistently upregulated in the aged mice, and the complement pathways are actually druggable pathways that we can aim to reverse or slow down the aging process in the cases that it might cause the diseases. And then we have identified the ligand receptor interactions that appear to play key roles in the aging process. And in the future work, we are looking at validating this type of findings in the human samples. So I'd like to finish by acknowledging my team. So it's the analysis led by Guyan, Feng, and Unju and Levi, and then the data generations by Yufan and Albert. And thanks for our collaborators. So for each organ, we have an expert domain to work on that particular organ. And thanks for the funders, and especially thanks for the BGI for the great collaborations. And thanks for your attention.
Do we have any questions for Kwan? Yes.
Is it on? Oh my God, it is on. Thank you so much, Kwan. That was really wonderful. I had a question about the integration. You said that you integrated with Visium. I'm curious, number one, like what bin size you needed to go for, given that it's sort of spot-based, but then StereoSeq is like can be really highly resolved, and whether the approach is sort of spatial aware or not?
Yeah, that's a great question. So we are still working on different strategies to integrate the two data type. But what we have done so far was, actually tested different bin size. So bin 80 is where the Visium and STOmics have a similar size of around 50 to 55 micrometer. And we see the two data types are very comparable in terms of genes detected and also the spatial pattern. But we are very interested in the complementary part between the two data type, and the single cell resolution. The bin 14 is the one that gives us a lot of interesting information about cell-to-cell interaction at single cell level. So the computationally, the way we are thinking of integrating these two data sets would be like to find the neighbor cells in the gene expression space, and then from there we hope that the cell type identification of the STOmics, which is supposed to be at a higher resolution, can inform the deconvolution of the Visium. And we also have a way to map the two adjacent sections so that we can actually map the single cell of the STOmics to the spot of the Visium, and that's another way to do deconvolution experimentally. So, yeah. Yes.
Hi, Kwan. Ranjini Thomas.
Yeah.
So the complement receptor and the complement 4 is really interesting. Do you see that the macrophages are close to the cells that express the complement, or they're far away? In other words, is the cleanup of the dead cells actually happening, or is there just inflammation being perpetuated?
Yeah, so we observe that across all 5 organs, but for the macrophage, we observe it's more in the spleen and the liver. Um, we suspect that complement factors produced in a high amount in the liver, and then it got transported to other organs. So currently, we are testing a very interesting approach that we haven't seen anybody try yet. That is to see which organ produce which ligands, and what other organs have the receptors expressed, and whether there's a downstream effect of that ligand transported to the distal organs that have the receptors, and that allow us to study the distal communication between organs. And complement pathway, I think, is the ideal pathway for that. Hopefully, we can see the signal from liver to the brain and to the peripheral organ like the heart and the kidney.
You might want to look at the gut. That's where ultimately it comes from to get to the liver.
Yeah. If you're interested, let's chat and yeah, thank you.
Thanks for a great talk, Juan, on a really interesting project. You've shown how it's really powerful to be able to sort of bin at different resolutions.
Yes.
I wondered how the segmentation approach that was mentioned by Kira that's coming sort of fit into the workflow.
Yeah.
And have you tried, the SAW approach and been able to get that to work?
That's a great question. Yeah, we try the SAW approach with the latest version of the pipeline. It works really well. And the advantage of doing cell segmentation is that we, instead of looking the bin 14, that we have a signal everywhere across the tissue section, now we have the signal where the cells are. So there's a lot of like empty space between two neighboring cells that has no signal, and that's accurate measurements. If we bin them, then we have a less resolution. The signal diffuse a bit between two neighboring bins. So so the segmentation is a good way to do it, and we apply the default the stereosync the SAW pipeline for segmentation, and it worked well. So we're working on the visualization of that, how to show the individual cell and the signal that map to the cell. Yeah. Thanks.
Great. Thank you very much. I'm always interested in research in diseases I might get, and I think aging catches everyone. So probably like a lot of people, I'll be Googling this afternoon how to upregulate my OXPHOS pathway. So now I'd like to introduce a rising star from the West. Dr. Jennifer Correnti is a postdoctoral fellow in the Sharma Lab based at the Harry Perkins Institute of Medical Research in conjunction with the Curtin University in Perth. Here they explore similarities between fetal and tumour microenvironments with respect to T cells in hepatocellular carcinoma, having recently contributed to a review in Nature Reviews Cancer and on oncofetal reprogramming and its implications in cancer development and progression. Furthermore, they are assessing the impact of oncofetal reprogramming within the tumour microenvironment on immunotherapy outcome, in part by employing several spatial technologies. Jennifer was invited to present their work on spatial technologies during a BioCompare webinar and is currently working with a newly developed StereoSeq technology to identify markers of an immunotherapy response in hepatocellular carcinoma. Jen's talk is titled STOmics Gen X, When StereoSeq Meets Jump Code.
Thank you, Richard, for that introduction, and especially a big thank you to Decode Science and BGI for inviting me to present this work for you in this lovely and early breakfast workshop. So these are my disclosures. So just in one day of Multiomics, we've already heard a lot of talks that really highlight the importance and the applications that spatial transcriptomics can have in research. And so these technologies really help to provide that spatial context within a tissue that you do lack with solely single cell technologies. And so it's a relatively new technique arising from around 2016 that has really expanded quite rapidly in the number of techniques available, as well as the number of publications that do cite these techniques. Yet with all of these new technologies revolving around spatial transcriptomics, there are a lot of variations within them. One of these is the field of view, or the capture area. So it ranges from quite a small capture area where you can only look at a very small section of tissue to currently the largest capture area, which is StereoSeq at the moment. The next is the resolution, so ranging from a low resolution where you are looking at several cells within one spot to a single cell resolution and also a subcellular resolution. But perhaps one of the most important aspects for finding new answering biological questions and also for the clinical translation is the detection efficiency. So you can see here on the x-axis several different spatial technologies grouped by their resolution. You can see that there is a difference between the detection efficiency for some of these technologies. Now, while our group does use several of these technologies, I'm going to be telling you about our work with STOmics. So Kira's already nicely covered this, as well as Kwang, but we have the StereoSeq technology on the STOmics chip. And I won't go through this too much, but I have included what I think and still think is a pretty cool video where you can see the tissue being mounted onto the chip itself. So here I'm holding the chip with forceps, and this is the newer version does have it on a slide, so just keep in mind that this is the original version that we're using here. The first version, sorry, I should say. And you can see the tissue section on top of the chip. So hopefully that plays. So you can see the heat from my finger does mount that tissue section onto the chip. And one thing I just want to point out here is that this is a poly-T, so it's a poly-A capture system. And as we've seen with similar single-cell technologies using this capture technique, there is, you do see a lot of ribosomal and mitochondrial content within this data. And this has led to some bioinformatic pipelines that try and reduce this noise when you are annotating and clustering cells. So this paper here was recently published in Nature just a few months ago, and it's really nice to exemplify in this paper how they have reduced the effect of ribosomal and mitochondrial content. So they have regressed out the percentage of mitochondrial data before their initial clustering, and then when they wanted to subcluster their cells, they actually completely removed 1,500 genes associated with mitochondria, heat shock proteins, and ribosomes. And so this raises the question, can you actually do this in vitro rather than computationally? So currently there are a few different technologies that try and tackle just this. One of them is Twist Biosciences. So this uses a targeted approach. It is probe-based, and they have existing panels available, but you can customize your own, and it is scalable. So you can look at a few hundred to several thousand genes. And a second option is targeted depletion. So this is your CRISPR-Clean from Jump Code Genomics, and they target genomic reads alongside mitochondrial and ribosomal reads and also non-variable genes. And this does have a customizable aspect as well. And so we wanted to go for an unbiased approach, and so we did go for CRISPR-Clean. So we wanted to combine STOmics with CRISPR-Clean to see if we can increase the detection efficiency. So you've seen this pipeline a little bit from Kira earlier, and so in green we have your conventional STOmics pipeline, and then in purple I've highlighted where we are bringing in CRISPR-Clean. So what we do is we have your tissue mounted on your chip, we take it through the pipeline, and then when we get to the cDNA section, we actually split it into two, and it either goes through your conventional STOmics pipeline, so through the green, or it goes through CRISPR-Clean. So you make your RNPs, you combine it with your cDNA, it undergoes your CRISPR digestion, it's then purified, and then we input it back into the library construction and sequencing. So what this means is that we have your STOmics and this new pipeline, which we have termed STOmics Gen X, or gene expression. It is the same tissue, and so the same spatial locations do match. And we are using 2 samples of liver tissue for this one here. Okay, so the first thing we wanted to do was to look at the impact of CRISPR-Clean on genomic data. So there are two samples, sample 1 and sample 2, and on the x-axis you have STOmics to the left of each and STOmics Gen X to the right. And then plotted on the y is the percentage of aligned reads. So you can see if we look at sample 1, about 84% of the reads align to genomic intervals, and 16% align to your transcriptome. But following STOmics Gen X, you see that there is an increase of 42% of these reads now aligning to the transcriptome. And a similar pattern is seen for sample 2. So what this is showing us is that with CRISPR-Clean, STOmics Gen X then allows for a reduction in this, in the number of genomic reads that are detected, and it also allows for a redirection of these reads to now be towards the transcriptome. And so after this, we wanted to know, does this translate to an increase in the number of genes detected? So looking at the library complexity, we downsampled each library to 1 million reads, 2 million, and so on and so forth. And we counted the number of genes that have 10 or more reads at each library amount. And what we found was that there was consistently a higher number of genes detected in STOmics Gen X, so these solid lines compared to the conventional STOmics pipeline, these dashed lines here. And so what this means is that while you also do detect more genes, you also can sequence at a lower level, which obviously saves you some money and allows you to run more samples. Yet the CRISPR-Clean technology doesn't only target genomic reads, it also targets ribosomal and mitochondrial content. So we next wanted to see if this is reduced. And what we found was a 2.3-fold decrease and a 2.7-fold decrease in the total percentage of ribosomal and mitochondrial content using STOmics Gen X. And if we look at this spatially, you can see that this top one, it displays the ribosomal and mitochondrial count in STOmics, and then you can see that reduction shown spatially following STOmics Gen X. So now that we have seen that there is a reduction in the number of genomic reads alongside mitochondrial and ribosomal content, we wanted to see if this increase in genes detected then translated to an improved ability to annotate cell types and clusters. So I'll take you back to this slide just quickly to remind you of how we analyze the data. So we do this in bins. So as already been explained, if you go 2 by 2, so including 4 bins, that would be your bin 2, because it's 2 by 2 spots, sorry. And as has been mentioned, bin 14 is roughly 1 cell, and the analysis from here on will be showing at bin 50. So we're looking at about 2 to 3 cells. So when we wanted to annotate the 5 major cell types, we found that we did actually find an increase in the number of bins that detected some canonical genes for these major cell populations, as you can see through this table here. So I'll just first take you through the major cell type of this tissue, being liver tissues, obviously your hepatocytes, epithelial cells. And you can see, this one. So you can see here that in STOmics and STOmics Gen X, you do see an increase in the expression of these canonical markers for this cell type. And if I plot that as a histogram, you can see that the UMI count does shift to the right, indicating a higher expression, and this maps to what we see within the tissue itself. If we go to the next most dominant population is our myeloid cells, and we found an increase of 1,227 bins that do detect some of these canonical genes for myeloid cells. And again, we do find that there is an increase in the number of bins with the UMIs mapping back to these genes. Next, if we look at fibroblast genes, there is an increase in 732 bins that were detected with fibroblasts. And again, if we look at the histogram, interestingly here, we do see that at bin 50, that UMI count of 15 and above, we only detect this following STOmics Gen X. So this is suggesting that this new combination of these two technologies leads to an increased gene detection for these canonical markers. So now that we have seen that there is a enhanced ability to detect these different cell types within a bin, we next wanted to know, is there any impact on the highly variable genes detected? So So when we looked at the residual variance for all of these genes in both technologies, which you can see on the x-axis here, we put in a cutoff of 1.4, and we annotated anything above that cutoff as being a highly variable gene. So what this showed us is that there were an additional 592 genes that were marked as highly variable genes following STOmics Gen X. And what's interesting to note is that of these genes detected, roughly 1,700 in STOmics and 2,300 in STOmics Gen X, there was some overlap in what was denoted as highly variable, yet between each technology there were some unique to each. So if we look at those that were only labelled as highly variable genes with STOmics, we found that 21 of these, shown in red, were mitochondrial and ribosomal genes. And what this plot is showing you on the x-axis is the UMI count for each gene following STOmics, and on the y-axis is STOmics Gen X. So it's important to note that we do actually detect all of these genes in STOmics Gen X, but they're just not labelled as highly variable genes. For the mitochondrial and ribosomal genes in particular, falling to the right of this dotted line, which is a 1-to-1 expression, indicates that their expression is higher in STOmics. If we look at those genes that are present as highly variable following both STOmics and STOmics Gen X, you see that their expression is a lot more concordant between the two. There are 1,015 of these that were detected in both. And what's important for us with our research is two of these genes in particular, so your alpha-fetoprotein, or AFP, and your haptoglobin, or HP. So these have some relevance in the clinic for hepatocellular carcinoma. That is that AFP is often used for HCC detection, and it can also predict recurrence and overall survival, and that's shown nicely by this plot here from this publication in 2018. So what they did was they took I think it was 81 patients with hepatocellular carcinoma who were about to commence chemotherapy, and they split them into either the non-decline group in blue, so that is where the AFP level was maintained throughout treatment, and the decline group in green, where the AFP level did decrease during treatment. And what they showed was that in the decline group, there was an increased overall survival, and they did also show this for recurrence as well. And in terms of HP, it has been shown to have lower expression in poorly differentiated cancer cells, and it also has several clinical implications across several other cancer types. So if we look at the spatial location of these, what we found was some spatially resolved hepatocytes. So we can see your AFP high and HP low, and your AFP low and HP high cell populations in a spatial context, which you would miss with single cell alone. If we look at AFP, we had an increase of 5,505 bins that had any expression of AFP, as you can see between the STOmics on the left and STOmics Gen X on the right, but this is a bit more apparent if we look at the UMI count as a histogram, because you can see a shift in the expression to the right, and similarly to before, anything with a UMI count of 17 or above was only present in STOmics Gen X. So we do see that increase and shift in expression. So just to summarize, we found that with STOmics Gen X, there was a reduction in the number of genomic reads that allowed for these reads to then be redirected to the transcriptome, which did translate to an increase in the number of genes detected. And there was also a decrease in the mitochondrial and ribosomal reads, and also their detection as highly variable genes. We found that there was improved detection of these cell type markers with STOmics Gen X, and that we also were able to identify some clinically relevant markers. So I'd just like to thank quite a few people. So this work was in collaboration with Luciano and Jasmine, and of course BGI and Jump Code for their support, otherwise this project wouldn't have been possible. And if you are interested in this work, we have just released this as a preprint as of Friday, so do check it out and read more if you are interested.